Hello friends, welcome back to my channel, and today in this video we are going to study about interference and coefficient of coincidence. Now if you remember from the last video, we worked on 3-point test cross and we obtained this mapped result which shows the gene C is between A and B and the distance are given. For those who have not seen the last video of mapping with 3-point test cross, I'll provide the link at the end of this video. Go and check it once before seeing this video. So this map distance, what information it provides? It provides the information about the proportions of recombinant and non-recombinant gametes in a cross. What proportion it is showing? Like between A and C here, 14.6%. This is the recombination frequency. Correct. 1% recombination frequency, that is 1 mu. So 14.6 mu means 14.6% of recombination frequency. So 14.6% gamete will be recombinant with respect to point A and C. What about with respect to point C and B? 12.2% of the gametes will be recombinant. Now for the double crossover probability, what we have to do? We have to multiply the probabilities for A and C and B. So after multiplication, we obtained why here it is written 0.14, 14%. When we talk about in terms of probability, we divide it with 100. So it is 0.14. So the probability for double crossover will be the probability of A and C multiplied with probability of C and B. So the answer is 0.0175. Now, this is the probability for getting the double crossovers. If you remember from the last video, what was our total number of progenies? It is 752. So if you multiply 752 with 0.0175, this much progenies should be of double crossover. After multiplication, our result is 13.17. Do you remember from the last video how much number of progenies we obtained for double crossover? If you remember, it is 5 and 4. That is 9. So the observed number of progenies were 9, but the expected is 13.7. So what we observed? The reduced number. Expectation is we should get 13 progenies with double crossover, but we got only 9 progenies with double crossover. Now why so? So this phenomenon is very common in eukaryotic organisms. According to this phenomenon, one crossover and another crossover, these two events are not independent events. They are dependent. The crossing over at one position inhibit the crossing over at another position which is located in the same or nearby gene. So the crossing overs in eukaryotic organisms are not independent event. They are dependent. The crossing over between one genes can inhibit the crossing over between another genes. Because of this inhibition only, we observe a reduced number of progenies. And this inhibition is known as interference. The degree to which one crossover interferes with additional crossovers in the same region is termed as interference. Now, one more term comes into play, that is coefficient of coincidence, which is the ratio of observed double crossover to expected double crossover. So, observed was 9, expected is 13, the number is 0.69. Now, from this number, we can calculate the interference. What this number is saying actually is that we are observing only 69% of double crossover. That means if the expected is 100, we are observing only 69 due to interference. Now the actual formula for interference is 1 minus coefficient of coincidence, that is 1 minus 0.69, that is 0.31. That means 31% or approximately 30% we cannot see due to interference. 30% of the double crossover, here you can see the 100, we are only seeing 69, or make it near to 70. We are only seeing 70. So that means 30% we are not seeing. So this 30% we cannot see this number because of interference. That means one crossover inhibits the subsequent crossing overs. That is about interference and coincidence. Now for this gene pairs, in the last video, you already worked on its gene order and the recombination frequency. These two you already worked in the last video for the same question. Now for the same question, what we have to do? You have to tell me the interference proportion. So that's all for this video. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.